So I'd like to thank the organizers, James, and all the fellows from Precision for providing me the opportunity to tell you a little bit about what we are doing. And before I start, I'd like to spend a few words about how do we define challenges in general in science, but then in general, really general. So let's assume that your car has fallen into the river, and now you want to take this car out of the river. What are you going to do? Call somebody is not a good answer. You have to think. You're now stuck inside the. Okay, so I'm gonna do it quickly because if not, I'm not gonna talk about science. So this is what you got. You have a pointer here. This one? It should be in the same one, guys. Technology driven. Okay, so what you can see here is a car that has fallen into the river, and 95% of the people that were asked, they said, you know, bring a bigger car. But only very sharp people or physicists actually see that this car is full of water, right? Therefore, the momentum that was calculated here is not even correct. And what will happen is this. Now, the problem is that you don't have one problem in the water, but two. What would be the classical solution? Bring a bigger car. And people just really do not learn. Okay? So these are real slides and came from Aarhus, from North Denmark. And you can see the lake here is actually an ocean, the Baltic Ocean. But this is a true story. So it's very sad. It means that, you know, we need to think a little bit out of the box. So I'll tell you a little bit about what we are doing, and hopefully I'll try to convince you that some of it is a little bit out of the box. Our lab is mainly interested in leukocytes-implicated diseases. So blood cancer, inflammation, viral infection, we are very much interested in different types. All those bluish diseases are very active program in our lab. But you can also see some solid tumors such as ovarian and GBM. They also have an immune component that is very serious. And we're also interested in rare genetic disease, real rare, that have maybe 3 or 4 examples, 4 patients examples. So really rare that leukocytes are highly involved. So our lab is kind of divided into 2 types of groups. One that are interested in doing drug delivery system, mainly from lipid-based particles, but we also generate monoclonal antibodies and fusion proteins. And the other one is doing more exome and transcriptome analysis. And the idea, of course, is to combine those 2 into one tailor-made appropriate vehicles, et cetera. And we are trying to decipher gene function mostly in the immune system. And the classical way was to go from in vitro cell culture into mouse model. And thanks for the work of Craig Mello and Andy Fire, we are able now to play around basically with RNAi. And we are trying to look from a drug discovery perspective into therapeutics, and we try to do quite a lot of work. I'm going to align here what are the challenges that we have in the field of leukocytes. Of course, there are classical challenges for everyone that you don't need any introduction, right? The efficient encapsulation of the RNA payload, the specificity. This is true for any drug delivery system, evading clearance mechanism, avoiding toxicity and immune activation. This is very, very important if you want to actually target leukocytes and subsets of leukocytes. Carrier internalization in the case of leukocytes needs to be very, very, very fast. Very fast. I'm going to spend some time on this. And then endosomal escape is essential. So before I'm going into this, just to give you a schematic illustration, this is, let's say, a lymphocyte. And lymphocytes, as you can see, the majority, about 80%, is a nucleus. So there is not really a lot of place to do RNA work. And if it's an activated lymphocyte, so you have about 70%, 75% is nucleus, and then 20-25% is cytoplasm. It's notoriously hard to transfect. And actually, primary T cells are hard to transfect, but primary B cells are even harder to transfect. And just a week ago, a paper from Dan Anderson group came in Advanced Materials looking at messenger RNA delivery with a new lipid formulation, or lipidoid formulation, for B cells. And it's a very nice paper, but at the end, the level of transfection in vivo is less than 7%. So viruses can do around 5 to 10% in primary B cells, but this is really calling for very specific ways to transfect them in vivo, and then, of course, in the clinic. So I'll try to talk a little bit about that if I could. This is very challenging, what's going on here with the timing. I started 24 minutes, now I have 28. That's amazing, right? And it's going up. Maybe I don't understand the system. Okay, so I want to tell you a few stories. One of them is an inflammatory bowel disease. A few years ago, we are very interested to understand why cyclin D1, this gene that's responsible for proliferation of both normal and malignant cells, is apparently upregulated mostly in leukocytes. And it was already shown that in epithelial cells, this is actually a very, very important target because it's basically causing epithelial colorectal dysplasia and calcinogenesis. But in inflammation, it wasn't really known. If this is the target, this is contribute to the pathogenesis of inflammation, it serves as a therapeutic target. So we set up a strategy to target gut mononuclear leukocytes and try to selectively silence cyclin D1 in experimental colitis model. The reason that we have done it was that there were several papers that correlates proliferation of leukocytes during inflammatory bowel disease with migration. Mostly T cells can migrate. And we understand that this is a problem, but the idea was that if we can hijack the idea, so basically just target those systemically with high specificity, and then deliver the siRNA against cyclin D1, we will be able to decrease the proliferation and therefore also the migration. Apparently, the hypothesis was completely incorrect, but it turns out that we learned something. So we published this paper about using this system. Then we didn't have the precision system, though, so it took us a very long time. We generated 24 monoclonal antibodies against beta-7 integrin, and those antibodies were labeled on the surface, several clones that we tried. And the good thing about this paper, not about the paper, but about the work, that we could show very high transfection under shear flow condition into those primary lymphocytes, where you put an isotype control on the particle, they are not penetrating. And this is a very fast penetration, about 45 seconds in human T cells, one of our clones, 1 out of 24 clones. The rest stick very nicely on the membrane. So take-home message number 1, you have to study the receptor interaction very, very fast with the ligands or with the monoclonal antibody you want to do, because not all of them internalize. Actually, the majority sticks very well on the surface and are not internalizing. And this is a classical example of one of the clones that is actually internalizing. And then, of course, the siRNA per se will not enter. And we have done the experiments also with a beta-7 knockout mice, and here again you see almost a black image. So under shear flow condition, that was very nice, but we eventually found that actually it's not blocking the proliferation and then for the migration, but it's actually influencing some of the pro-inflammatory cytokines. And that was really a stellar discovery, and that was based on cyclin D1 and not D2 or D3. So it's very, very specific to D1. And you can see that, again, this is blocking the intestinal inflammation. nicely when you have those cyclin D1 siRNA, and more is in the paper. It's an old paper already. So we basically target this into this high affinity area of the ligand binding domain, and one of these antibodies, one of the clones is entrolizumab that is now being in final development in Genentech and hopefully will be marketed by the end of the year. I don't know what's going on with all the results, but veloduzumab is another antibody also targeting the same integrin, but this time with an alpha-4 beta-7 integrin. It's marketed by Takeda, and it's going into a different area in this joint alpha-4 beta-7 integrin. Okay, so what are we doing with these antibodies? We also do molecular imaging, basically looking at disease management using PET-CT, and you can actually do this on a precision system as well. You can do your chelator, so you can take the antibody, and I can tell you that at least one of the clones that we're using gets beta-7 integrin is not susceptible to anything, basically. So you can inject it into one of the ends and then do your chelator conjugation, and then you have to purify it. But basically, we have shown that you can actually go in vivo with this and see very nicely the gut area. So this will be a very nice platform for disease management during IBD. Now I'm moving to a different disease, but I think you will find it with some common areas. So mantle cell lymphoma is a very aggressive form of B-cell non-Hodgkin's lymphoma, quite rare, about 6% of the non-Hodgkin's lymphomas. It's considered to be incurable, including with a combination of rituximab and chemotherapy. Now the median survival increased dramatically from 2 to 3 years into 5 to 7 years, still poorest among the B-cell lymphoma. But it has some very unique genetic profiling. This disease has a chromosomal translocation that the gene that encodes for cyclin D1, CCND1, juxtapose from chromosome 11 to chromosome 14 before a very strong promoter, basically the Ig heavy chain promoter, and start to produce a lot of cyclin D1. Now, B cells proliferate in the body, but they're proliferating through cyclin D2 and D3. Very rarely, cyclin D1 is upregulated. And if it's upregulated, it's usually means that something wrong is happening. And so in mantle cell lymphoma, 85% of the patients have this translocation. In multiple myeloma, 25%. And in other B-cell malignancies, it depends. But this is usually a bad sign if you have an overexpression of D1 cyclin in B cells. So what we have done, together with the help of guys at the Broad Institute, we sequenced around 81 patients, and we revealed 2 transcripts, a short messenger RNA for cyclin D1 and a long one. And then the idea was, can we create a strategy that will overlap those 2, and can we knock down both of the transcript? And together with Alnylam, that helped us a lot, we screened more than 500 sequences of cyclin D1, and we published a sequence. We found one of them that is really therapeutic sequence at the picomolar concentration, very potent. It can silence the gene. It can inhibit the growth inhibition, cell death, of course, cell cycle arrest at the right place, etc. And then, this is a way that you can tell your graduate student, Shiri Weinstein, now an MD, PhD, but back then a student, PhD student in my lab, that if you want somebody to leave the lab, that's a great way. Just let them screen 500 sequences, electroporate everyone, and then see what's going on. And then, 3 a.m. in the morning, she's trying to send emails with results. So definitely, it's really a challenging work. Then a French guy came to Itai Antoine Toker, and he also, together with Shiri, I asked them to develop this highly selective particles. Can we do something to encapsulate this one sequence? And we found out that we can do it. The only problem, there is no animal model for mantle cell lymphoma. All the models that we have seen or read are basically taking the cells and implant them subcutaneously. So this is not a great model. So we had to develop one. It took us about 2 years to develop the model. Last year we published this and other things in PNAS, and basically it's a skid, a very special skid model, and you can implant, the engraftment is fantastic, a human mantle cell lymphoma. These guys are transfected, stably transfected with GFP, but you can do any one of them. And after 1.5 hours, you will find them about 32% already in the bone marrow. So it's very, very specific. You have a little bit in the lung stack, almost nothing in the liver or kidneys, nothing almost in the spleen. And again, 1.5 hours is very short. So they really know where they're homing. 24 days later, you have those huge lesions inside the bone. And they will die between 24 to 30 days. Very aggressive model. Not sure it's exactly the human equivalent, but if you test drugs that have been approved, like ibrutinib on this model for mantle cell lymphoma, it works well. So apparently it has some kind of a translational gap, and we like this model. So we were looking for choosing a receptor target, something that will be internalizing our particles very efficiently. And as you can see here, this is a screen that we have done with different targets, LFA1 and other integrin, alpha L beta 2. And actually in T cells it works very well, but apparently here the expression varies, and this is CD22. We chose CD38, and we saw that Sanofi has an antibody against CD38, and this was under a phase 2 clinical trial, very successful. We looked at the clone. We found one clone that they claimed was not therapeutically relevant, but could be used as a targeting moiety, and we used it. I don't know if you can see here, but actually, when you inject this antibody labeled, you can find them, again, 1.5 hours later, about 42% of the injected dose goes to the bone marrow. It's double positive, so you have a CD38 and the GFP of the cells. Compared to almost everything else, it's very clean. And then when you sort the cell, label them with anti-CD20, you can actually find them internalizing. So this is the complete antibody. Then we thought, okay, now we need to formulate the particles. We use ionizable lipids to formulate in the precision system. And the reason that we use ionizable lipids was because of this paper that we published in Biomaterials in 2010. And if you're not familiar, this is a very important paper. Actually, this paper found that permanently charged amines can cause immune response by, at least in lipids back then, by targeting toll-like receptor 4, and then induce an NF-kappa B signaling effect. And that could explain many of the adverse effects that is happening, like cytokine storm, interferon-responsive genes that are going up, et cetera, when using permanently charged amines. It doesn't happen if you do an ionizable system. So it could be polymer or lipids in this sense. So we see it much less. And we proved this with a TLR for knockouts. Later on, people have published also TLR2, which is very similar. So I think it's important to use ionizable lipids. So the things I'm going to show you today is based on ionizable lipids that Peter Kulis and the friends in Vancouver developed. But we have our own lipids as well that are now being studied. So we do this on a precision system. Then we take a reduced antibody, and we run it again in the system. And I think this is a very nice application because you can do 2 runs on the same chip. So, and then we get a nice formulation. So we publish it in a few journals and for different indication. This is from the PNAS paper with an anti-CD38. You need a colon to wash it back. So this is a gel filtration chromatography. And then, you have the removal of the unbound monoclonal antibody. You need to control the orientation, which is another very challenging thing. We have a paper coming, hopefully now in Nature Nano, which we try to solve it in a biological way. So I'm not going to talk about this because I don't have time, but just in general to show you some data on this approach with an anti-CD38. We use a co-culture system. We took cells that express the CD38 and cells that lacking it. We show competition with a free anti-CD38, and it looks very specific to this. We also use patient samples, and it's also very specific. And then we did a shear flow assay. Again, you see that those anti-CD38 particles that have an siRNA, this one is 647 Alexa, are entering the cells where isotype control are not, and the uncoated particles are not entering as well, not delivering. We found that it's really nice that they're blocking the cell cycle in the appropriate area, and we did a very important experiment blinded in Charles River when we look at survival. So here you can see that two things. First, a single siRNA is not enough. Although it's extending the survival dramatically, it's not enough. It's a very aggressive model. These are the mock-treated. But you also see that this antibody that allegedly didn't have a biological activity do have something, because it's extended. This is an siRNA luciferase, and it's extending the survival as well. So this is a platform, and it can actually, by changing the antibody, and again, this is the orientation thing, you can actually be in a very good position. So I want to spend a few more words about another platform that we're using also on the precision system, which is the Gagomers system, and this is a collaboration with Quiet Therapeutics here in Boston. And the idea here is to use the lipid drug core matrices and then hyaluronic acid on the surface. These are lipid-based nanoparticles coated with high molecular weight HA, and there is a variety of questions what you can do with this, but you know, CD44 as the receptors is highly expressed in many types of tumors. It has an alternative splice variants, about 10 of them, and they all change between the stems area. And it's an adhesion molecule. So the interaction between the natural ligand and the receptors is very similar to integrins, other adhesion molecules. And we have shown this for many years, almost 20 years of papers on CD44-HA interactions in tumors and in other cell types. But basically, you know, this is a wide spectrum thing, and I'm not going to talk a lot about this, but we have lots of knowledge about the area that is the ligand binding domain. And we think what is happening is that the receptor needs to be tilted in some way to bind in high affinity to the natural ligand. And then some of them, depending on the tumor, will internalize. Some of them will not. We published heavily on this from many years ago, even before 2010. 2004 was the first paper. Then with paclitaxel, a paper in Biomaterials, another one in mitomycin C, doxorubicin, nucleic acid, et cetera, et cetera. I want to share with you some selective targeting to tumors in vivo using this system. And again, it's running on a precision system, so it's a nice thing. This is a B16 melanoma, sub-Q tumor inoculation, single IV dose of Cy5 labeled siRNA, 2 mg per kg, it's quite a lot, into the tail vein to tumor-bearing mice. This is fluorescent spectral, basically in confocal microscopy and 4-micron cryostat preparation, 6 hours post-administration. This is the tumor. You can see it's full of siRNA. This is the liver. You can only see in the sinusoid and the blood vessels, but you're not seeing the hepatocyte. This is the spleen again, and this is lungs. And this is the capillary area. So it's very, very interesting that it's accumulated in the tumor. And this is just some quantitative data about relative quantitative of the siRNA. You can see knockdown in the tumor about 80%, and then nothing in the liver or lung. This is another work of Quiet that they're doing with diphtheria toxin A expressed under a CMV promoter. And again, this is DNA delivery, this plasmid. And you can see that these are all different controls. But at the end, few injection, last treatment, it's lasting for a very long time with tumors that are already established. This is a nude mice of human prostate adenocarcinoma, it's PC3, an IV injection at 2 mg per kg. Okay, so just to summarize this part, is the Gagomers platform. It creates new class of proprietary, et cetera, et cetera. RNA platform is a broad area of novel cancer drug, and the small molecule could be also worked well. I want to say a few more words about a local delivery that, again, we did with hyaluronic acid, looking at CD44-HA interaction with glioma. This is mainly the work of Srinivas Ramishetty, also a T-cell guy that is doing lots of T-cells activity in my group. And here, a friend of mine who is a neurosurgeon came to me and said, listen, we have a very unpleasant situation where basically patients, the operation is successful, but the patient will die in 12 to 15 months. Can we do something? Can we inject them locally into the tumor bed? And I said, what should we inject? And he said, let's find some targets. Let's see if we can do something to help those patients. And we were looking for glioma unique surface markers that can be used. And one of the things that we surprisingly found is CD44. So CD44 is also expressed in glioma cells. And these are patients, different patients. This is the expression level. They are all pathologies as this. These are cell lines that are classical U87, T98G, U251. They're all highly expressing pan-CD44. And again, we run it on the precision system. So we develop basically particles that have on the it's crashed. I didn't do anything. Okay. Great. I have to start again. This is the gut microbiome, actually, what you've seen in the picture. It's an electron microscope that we have done, not this one. More. Slide 30, 47, 48, 49. That's good. Okay, so we basically run on the precision system, ionizable lipids, the classical MC3 lipid with a classical formulation, I would say, everything was published. And then we encapsulated PLK1, a pore-like kinase. And the reason that we have encapsulated pore-like kinase was that actually most patients highly express this in glioma. And we did a model. Yes? Okay. We did a GBM model, orthotropic model, basically injecting into the mice. You can see the tumor is very focused, so it's not a classical GBM, because in GBM they're already spread around. We have a collaboration with Indoverma on a syngeneic model, which actually spread seriously, not only solid tumor, but really lots of things. And I hope to be able to tell you more about this next time. And what we have found that actually we can get a very efficient silencing on a single administration. We administrated tiny amount into the tumor bed. This is done usually under MRI, and it's very challenging because if you inject more than 5 microliter into a mouse brain, you will get edema. So it's going to destroy your experiments, and of course it's going to kill the mice. So this is something we don't want. But we found that after 4 administration at day 7, 9, 20 and 22, we get this survival, which is really amazing with SI-PLK. And again, this was published. So the challenges that I wanted to talk about in selective targeting is first avoiding mononuclear phagocytes. Now everyone knows what this, if this is a nanoparticle, what is this? This is PEG. Okay. So it's a British guy, of course. Okay. And then high specificity and functionality. This is, again, If you align your targeting moiety in a way that you can control the orientation, and hopefully next time I'll come, this will be the paper I'm going to talk about, is the orientation that we can control biologically. That is phenomenal. And then the fast internalization and burst release, in the case of nucleic acid, is really, really important. So either you can facilitate the internalization, and then there is a recycling process, like in integrins, not in all of them, but in some of them. Or, you need to be able to deliver your payload very, very quickly. And it's not correct only for leukocytes. It's actually correct for many other cell types in vivo. This needs to be a very fast process. And we don't really understand enough the biology to put our finger and said, it will work with this and it will not work with this. The example I gave you initially with an anti-beta 7 monoclonal antibodies is that we have developed 24 different clones. Only one of them internalize very fast. The majority are not internalizing. And we had to screen this. So they sit very nicely. They stain the membrane very nicely. And often people said, you need a high affinity protein, high affinity antibody. A very high affinity antibody will not internalize. In many cases, it will stick very nice to the membrane. Actually, you don't need a very high affinity. You either need to have a good avidity that you can control, or you need to be able, and it's a matter of luck, to go in the same direction to the natural ligand. So if you do a co-crystal, if you have two proteins and you can co-crystal them, the natural one, there is a way that you can actually engineer this to look at the ligand binding domain. And these, in the case of adhesion molecules, are mostly internalized. So LFA1, ICAM1 has been investigated for many, many years. Believe me, I personally had a few papers on this during my postdoc. And if you look at other receptors, it's really depending on the interaction and on the specific clones. This is true not only for monoclonal antibodies, but this is true also for ligands that are sugar-based, like hyaluronan or hyaluronic acid. Okay? It's not going to be a solution for everything. So in a naive way, I can always tell you that we solve all the problem of everything. But the reality is that it's much more complex. And the targeted systems, in order to be advantageous compared to a non-targeted system, needs to have a defined property. In this property, the receptor-ligand interaction is of great essential. And if we were not able to study those in a specific context, in a specific setting, the chances that it will beat a non-targeted system is very, very low. And there are many papers over the past few years that said, targeted systems are less efficient than the non-targeted system. And they might be correct. It depends on the system that were tested. Okay, so if I'm now going to have a paper on those 24 clones, which is a boring paper, okay? 24 clones of anti-beta 7 integrin on a liposomal surface, and I'm going to show you that only one of them internalize and deliver the payloads, you might learn something or you don't learn anything. And what you can learn from this in general is that studying this in the appropriate context, for example, in vitro under shear flow condition, is probably the only way to find one specific clone that can do it. And if not, this strategy will never work. But if it works perfect. So I don't know. My dream is to be able to come in a few years saying, because of the precision system, we are now able to study it easily and robustly for a very, very long time, and we have some solution for specific cell types and for specific ligands that fits to the specific indication and the specific type of diseases. At the end, I want to thank many people, but the majority are the people from my lab that is really doing the work. It's a mixture, international mixture between people that are chemists, biologists, engineers, physicians, et cetera, and the funding agency that keeps supporting us, which is fine. And the last slide is, you can see that the only guy who has here a glass of wine is me. And everyone always asks why. And I said that everyone is working. I'm only one that is, talking. So I think that because of these guys, we are surviving and we are making progress. And I keep on chatting around. So thank you again for the opportunity. I'll be happy to answer any questions.
Number of questions.
Yes.
Should be on. Great talk. So you developed 24 clones and just one internalizes rapidly. Is it specific in that model? Because you just mentioned that it depends on the system. It seems to be overall general?
So we tested those 24 clones against the beta-7 integrins. These are originally rat anti-human cross-reactive with mouse. So we, human cells and mouse cells. And the one that is really internalizing is the one that is internalizing in both human and mouse, across those two. This is not the drug. The drug is an antibody that has high affinity. It's going actually for a different reason. It's blocking the homing of the lymphocytes from the circulation into the gut. And of course, it's a humanized version, et cetera, et cetera. So it's not this drug. But again, this screening, this cell biology, I call it, is essential. Because without this, it will be very hard to compete with any other system, with any other approach. And again, the amount, for example, in B cells, transfection is very, very low. So only when we put an anti-CD38 antibody, and we had luck because this clone of Sanofi is the clone that actually internalizes. But it doesn't have an ADCC activity. So for them, it's junk. For us, it's gold.
Okay, any further questions?
Got a question from one of our Twitter followers, G. Hain. Is this lack of targeting specific to antibodies, or do you think it's true of peptides and other strategies?
Everything. But I must say, again, you need a recognition site that is big enough. So it depends. If it's a tripeptide, it depends on the affinity, really. But if it's a longer peptide, it's almost a protein, it's a different story, but it will happen anytime. Again, this is for a systemic administration. I'm not talking about local delivery here. So this could be a completely different story. The barriers in systemic administration, except from anything else, the immune system, the clearance, etc., is to be able to target your cells, and then the particle needs to either internalize and release the payload fast, or that the interaction between the receptor and ligand will create a conformational change in the particle that opens up and releases the payload, which is kind of a science fiction if you think about that. But the reality is that if this happens, it needs to be a fast process. Because in culture, it could happen anyhow. If you put particles on any cells, just a matter of time, 24 hours, 40 hours, 48 hours later, you will get response. You will get your payload inside the cells most likely. Okay? Some of them will be exocytosed as well, but it will happen. But this is not the right system to investigate this. And it gives us a lot of negative, or I call it false positive actually, in this sense. But it's not a target, it's really not really a real targeting effort.
maybe time for one more question.
Thank you. So in terms of targeting with a specific peptide or antibody, and you said speed is really important, do you see differences in the mechanism of internalization, if it's caveolin, clathrin, or micropinocytosis?
Of course.
And do you have any idea?
I need
for which
another
is-
talk for this, but yes.
Okay.
Yes. So it's a perfect question. And the reality is that if it's a clathrin-mediated or caveoli-mediated or lipid raft-mediated or non-caveoli, non-lipid raft, etc., it really depends on the ability of your particle to release and escape as fast as you can. But even if you take proteins, as an example, that do home into the lysosomes, okay? For example, glucocerebrosidase, an enzyme that is used for enzyme replacement therapy in the case of Gaucher disease. Even then, if you incubate this, and they have sometimes they have a mannose-6-phosphate end, so the mannose-6-phosphate receptor will take it up, and they go to the lysosome. If you quantify this, you will find that it's a small amount that is actually reaching the target. In percentage, a few. And the reason is, can we make it better? And I mean, there is biological reason behind this, but can we improve this? I believe we can. It's just a matter of the formulation, the kinetics, and how much time and efforts and financial resources you're going to invest in this. But I'm in academia, so I can spend my time in playing around with this.
One question here. In regards to using antibodies as your targeting agent, are you looking at antibody fragments, or is there any reason to use one variation than the other?
So it really depends, you know. The advantage of small fragments usually is for, if you think about cancer as an indication, is that tumor penetration should be better. However, in the context of nanoparticles, I don't see a reason why it would work better than an IgG. Okay, and IgG has its own problem as well. But if you can protect the FC portion from a cost-effective standpoint, it's easier to make an IgG than a single chain or diabody or minibody or whatever. Much cheaper. And then if you have a method to hide it and control the orientation, again, I'm revealing a little bit of what we have done, but basically, we have done a biological instead of chemical conjugation. If you do this, I think that, you know, all these challenges, tumor penetration will still occur. But most of our targets are circulating cells, so I'm less concerned.
Okay, well, I encourage you to bring your questions to Dan at the break. Let's thank Dan again
Thank
for
you.
that great talk.